to a somatic cell or the body cells. So how is it different from the different kinds of like basic natural reproduction? So you see that the organisms, any kind of organisms are produced through a couple of methods, which are your asexual method of reproduction or the sexual method of reproduction. So when we talk about the asexual reproduction, we are talking about your basic mitosis, which will give rise to cells. This is seen in your somatic cells. Whereas the germ cells that are present, you will have the sexual reproduction, wherein the embryo from the female, the egg from the female and the sperm from the male is going to be, they are going to come and, they are going to be fertilization that is going to take place. And this will produce the first zygotic or diplotic cell, which is the zygote. So you will have a reduction division. As you know, it's meiosis is a reduction division. So haploid set of egg and haploid set of the sperm will come and fuse together to form the diploid zygote. And this is taking place inside the, what do you call it, the basic, the chromosomes are from both the male and the female, especially in the case of the progeny. So that is where the newborn is going to get half the genes from the mother's egg and half from the father's sperm. In cloning, what is going to happen is the egg nucleus that is formed is being removed. The nucleus is removed. So you're going to get an enucleated cell. This is taken and replaced with the donor's nucleus. And then this is going to be, the criteria for selection is based on the kind of properties the donor is having. Obviously, we want good breeding or good, the donor should be excellent in health or different kinds of traits for this particular process to occur. And then this is taken into the egg cell of the, egg cell. And then this is supposed to be again implanted and then grown. That is why you're going to get the clone of the donor. So you can see meiosis, you're going to get egg, sperm cell, fertilization. This is the single cell embryo. This is the 3-day embryo, the 5-day embryo. Over here, what you're doing is egg cell is present, removing the cell or the nuclear material, the nucleus, and then you're going to take the nuclear, from the donor cell, you're going to take the nuclear or the nuclear material. So this will have the chromosomes. So since the chromosomes are the genetic material, inside when you put it into the cytoplasm of the recipient, the donor nucleus will have the whatever progeny is there will be resembling the donor, not the recipient. So cloned embryo is going to be genetically identical to the donor. So history is basically sea urchins were isolated, the blastomeres of the sea urchin were isolated, and then the separation of it into 2 large cells. This got matured into adult organisms. Again, later, Dr. Hans Spemann tried this experiment on salamander embryos, and he produced twins. And 1958, Stewart could get cells from roots of carrot and was able to create a basic clone. But later, what happened was in 1984, Steen Willadsen, he tried to clone sheep using the DNA from the previous embryonic cells. Later, he could clone a cow from 1-week-old embryo cells. So just the embryo cells were taken, the nucleus was taken, and then the clone of the cow from the donor cells were created. Then in 1996, that was the game changer when Dolly was born. It is a sheep, wherein adult cells, somatic cells were taken, and then from there, the cloning was done basically by Wilmot and Campbell. And now after that, you have a lot of clones that were produced. Now, what are the different types of cloning that you can get to see? You have DNA, RNA cloning also. When you get to see DNA cloning, what is happening is all the DNA fragments that are there in the genome, first is restriction endonuclease nucleases are made to act upon it, so you get DNA fragments. Then later, it is cloned randomly, and the vector for this cloning is going to be the phage DNA or plasmid. And the term genomic DNA clone or chromosomal DNA clone then actually refers to the cell that is carrying the cloning vector. So that the phage isolates with a specific DNA insert. So now you can actually see this particular process how it is done. Right. Cloning vector and then linearization. So you have different kinds of restriction endonucleases such as right? BAM, H, and HIND3, such ones, what do? They cleave at specific sites so that you will get fragments, either sticky ends or blunt ends. After that, what is going to happen is you have cellular DNA again. Same restriction endonucleases are going to be acted upon by the same amount of DNA, so you get the same complementary sites. Then linearization with the DNA over here inside the cloning vector. Then this introduction of vector with the different DNA inserts into the suitable host cells. So this is how gene cloning or DNA cloning is done or is done, and production of the genetic duplicate of the existing cells. So now a human clone is a person who has complete set of DNA same to that of the donor. Twins are not clones. They have certain identical features known of, but there is a certain difference in the genetic makeup. That is the reason why twins also do not have the same set of fingerprints that are present. Your fingerprint is unique to yourself. So that is the reason why even your what do you call it? That is the reason why twins are not clones. So reproductive cloning has been having a lot of ethical debate going on regarding it. Reason being, is it necessary? Why are human clones needed? And what is the psychology behind pathology like children are there? And why are we playing God? And then what is the genetic? Why do we have to genetic? So basic ethical concerns, social concerns are there. So finally, Dolly was one such sheep that was produced as a clone, and unfortunately, after a some time, died. So how was it that they could generate Dolly, or how was it that they could create Dolly? So udder cells from the udder of the sheep. So the sheep was taken, and then they were cultured, and then to culture them, their genes, some of the genes were what do you call it, made silent or switched off as it is known as, and then unfertilized egg cell was taken. Then they removed the nucleus from the egg cell. Then what did they do? From the donor cell, udder cells, they took the nucleus, they inserted it into the egg cell using electric pulses. And then once this happened, they gave it two pulses. First one for fusion, second one for cell division to start taking place. And then you had an embryo that was formed, right? So this resulting embryo was taken and then implanted in another sheep, which is the subciliary. After gestation, the mother gave birth to the baby, which was identical to the udder cell code. So this was how Dolly was created. Now you have reproductive cloning. What are the benefits and the risks? So you need reproductive cloning for the production of higher breeds of, especially we are talking about animals. So you need whatever animal which is able to give more milk. When you do a clone of it, that clone also will be able to. We have this basic concept that it will also be able to give more milk. That is the reason we are cloning. Or in case if it is for livestock, that is livestock example. In case you want a better quality of meat available from that particular animal, you create a clone of that particular animal, then you're going to get the same quality of meat in the other clone also. So it maintains the good DNA, and then cloning genetically modified animals is like, if we do that, we can avoid transplantation rejection reactions. So example is your xenotransplantation, in fact, and you can also produce insulin. But there are certain risks that come along. First is it is highly inefficient. We do not know whether this will is feasible or not all the time. And then why is it that the embryo is not able to be sustaining inside the up to the stage of implantation also? And of course, to create such kind of fusion, you need a very highly equipped laboratories, and the infrastructure is going to cost because of the sterile conditions and the aseptic conditions that have to be maintained, as well as it's going to cost you a bomb just to maintain the plant. How right is it to experiment on the animals just for our necessity? And what if we do human cloning? Can you take your clone, since it is your clone, and put him or her in the same other class when you are in another place? Is it ethically right? Will your clone also be able to lead a proper life just like you are doing, or like a life just like you are doing? Will the clone also be subjected to the same kind of pressure, and at the same time, same kind of ailments? So yes, it is ethically, morally very questionable. That being the reason why we do not have much of human cloning as such. You never know what is going to be there in the future anyway. Now you have embryonic stem cells, which people are trying to see, especially the researchers. I should use the word researchers, wherein they're trying to see whether it can be used for therapeutic purposes. So, if at all there is an organ that has been completely destroyed or damaged, can we make a clone and then replace that particular organ? Or in case if at all there is any kind of injury that is taking place, can you regenerate? Or if at all it is too much destroyed than being able to be salvaged, then is it possible? So again, benefits are you can produce whole organs from the stem cells. So you can do like again healthy cells for the transplantation. You do not need to be on the wait list for having an organ transplant, or you do not need to wait for a person to die in an accident, or that is how the wait list process will be there in the first place. So for this organ donors to come up, or you do not, don't need to test different drugs or understand the diseases as such, or because it's a clone that you're creating. But then, having said all that, you have to remove the cell nucleus and you have to denucleate the embryo, and it is again a big process. So you have to keep them. So somatic body cell having the desired genes, denucleated, egg cell, nucleus removed. So from the egg cell, you are removing the nucleus over here, having a haploid cell. Had it been fused, it is a different story altogether. But for your purpose, you're taking the nucleus from the somatic cell and putting it into the egg cell nucleus, and then preparing a clone, which you're going to see that you're going to put it or implant it in the surrogate mother. For therapeutic cloning, you're going to do for tissue culture. So this is how you will have basic cloning as such. Next is home human cloning, so that we will be disease free. This is what we think we can. Is it possible? So the process of cloning, how it is going to be. Again, same thing, somatic cells and all that. Now we have the animal cloning. Unfortunately or fortunately, human cloning is still going on. The ethics and the morality of it is being questioned. So when it comes to animal cloning, people have started cloning their pets so that they will not have this particular they'll not feel bad if it has died because of some causes. So first cloned cat was born in 2001. Of course, we are like years later we are talking about. So here what you can do is you can take a biopsy, liver cells biopsy of the animal that has recently or very say in a couple of days before a couple of just as soon as it has died, tissues are grown, then embryo is taken, cloned, and then the genetic material is that is the nucleus is taken from the eggs from different cats, fused together by using electric impulses or electric impulses, then have a resultant embryo implanted into cats, so that you can induce an artificial reproductive cycle also, giving it hormonal injections. So, but then it is up to the cat whether that pregnancy will last or not, because again, because of the different kinds of conditions that pregnancy has to sustain or be sustained in, so that risk is always good. So next is a puppy that was cloned from a mixed a female dog, and DNA from the skin cell, that is ear cell, was taken, fusion, growing egg was implanted Africa surrogate, and then you have an Afghan clone puppy born. Because it is from that Afghan clone puppy you take it the skin cell. Of course, we have seen this donor egg from fusion and white-faced sheep. The egg is being given, egg cell fused embryo. So for the white-faced sheep, you're getting the black-faced sheep, white-faced lamb. Advantages. Because of this, we can be sure that you not run food fodder or milk. And so this is one of the advantages wherein improvement of the livestock. But then for human cloning or for cloning, people believe that we should not take yogurt, we should not do such kind of experiments. So we never know how things will turn out. The ethics are always there. So you can get the same kind of pet had already. Organs can be cloned, there are transplanted so that people can live for much of it is feasible, how much of it is possible, that has to be seen. Ear has been successfully or surgically attached after, and also there are genetically, so it is safe to eat the animal. So, but then usually, whenever you see, whenever the offspring of the cloned animals, the progeny of the cloned animals are often born with certain kind of deformities, because it is not the same genetic makeup that we are facing. And certain people do not believe in this religiously, and they do not, the pets that are actually clones of the donor nucleus may come or may not come in the same manner that you are expecting, since having the same kind of personality also. Risks, again, like I tell you, it is basically expensive, inefficient, you never know when it is going to be a failure. 90% of the clone attempts, they see that there is a viable offspring which is not produced, and so low success rates. And these cloned animals could not get acclimatized to the surroundings and they were not able to live for a longer time. And looking healthy at a young age itself is not a good indicator of whether the particular animal is able to survive long term. So for that is what they have given. Australia's first cloned sheep looked very healthy and energetic. And then finally, when they did an autopsy as to why the particular sheep passed away, nothing came. It was an inconclusive result. So where is this cloning that can be applied? So you get to see that cloning is applied for animals to be produced as drug producers. You can see bacterial cloning, also bacterial cell division actually helps a lot because of the production of insulin. Whereas animals can be used as drug producers or models, breeding or antigen tissues and for xenotransplantation. So, so you can see that in livestock breeding and agriculture also you can produce. So the meat from the milk from cow, pig, and goat should be, or they have been declared as safe. So this is all for references part. So different
Yes.
kinds of cloning vectors are your especially for DNA vectors you have different kinds, that is your cloning vectors. So it's a small piece of DNA that is taken either from plasmid virus, and then it is able to maintain its structure. Most of them are going to genetically engineered, and it is and it is chosen as per the kind of DNA, the size, and what is the type of DNA that you're using to clone. So what are the features that are there? Should be able to treat the foreign DNA and the vector along so that you get the same restriction into place, and then the same type of ligase so that they will be able to vector. So why is it necessary? So because this vector is the vehicle that can carry the artificial or the foreign DNA, and this can be amplified also so that you get many copies. And these cloning vectors are those that are able to transport the DNA that is there from the biological to the test tube. So you can see over here, plasmid is taken from the bacterial zone, and you're using restriction endonucleases to cut the plasmid. Over here also cell with the DNA is there, that is taken gene of interest and cut same DNA, and then this is going to be ligated using ligases or DNA ligase. That is. This is how you're going to get the recombinant DNA plasmid, and this is going to be inserted by transformation method into the what do you call it, the bacteria, and then the bacteria should be reproduced. Like since like there should be bacterial separation taking place, so you get a clone of cells. So this is how cloning vector started. So you have a Boyer's lab scientists working for research in 1976. You had first plasmid that was designed. What was a PBR plasmid polymer and powder plus number 320. So around 4 kilobytes in size, and it had system. This is how you had 60 genetic combination. 70s you had restriction enzyme and enzyme mapping of cement 40 virus. 68 DNA ligases was first isolated, and then 74 you had Swiss like sense, and by the time it was 78 you had discovery of the restriction into places that take place. Isolation was done in 1970, but discovery of restriction endonucleases was given the Nobel Prize in the year 1978, and first cloning vector TPR322 was given in 1977. So you have for a cloning vector, this is a multiple site. You have a resistance gene that can be inserted. DNA origin of application can given. So you have an poly sequence over here and specific type sequence that is there at the cloning site.
Excuse me, ma'am. Ma'am, what about the bacteriophage, ma'am?
I'm sorry.
Bacteriophage.
Bacteriophages are the viruses that can take in. What do you call it? This is the bacterial plasmid. So phage DNA is again different, and that is happening in the case of the plasmids or the bacteria that are taken up the virus taken up by the bacteria. So that is making it called being called as the bacteriophages. So that also is able to take a little bit amount of DNA into it. Okay, that is also a cloning vector which can be used. So now we have to have a selectable marker over here, confers resistance to certain amount of antibiotics that are there or the selected agent. So what is happening is, whenever a cloning vector has this particular marker, it is able to survive in the hostile conditions against that particular against that particular antigen so that this antibiotic acts. It is not going to die. So then we have something called as the reporter gene or the marker genes, which are used in the cloning vectors so that you can see which ones are the successful clones. So just the different kinds of color and trypan blue staining, such kind of methods will be able to know. So you can put some reporter gene or marker gene. Then additional properties, of course, they should be small. So you have bacteriophage that is there, like you have asked, and then we have our plasmid DNA. Then you have cosmid. Then you have yeast artificial chromosome. These are all different kinds of vectors wherein they can take up large quantities, small to large quantities of foreign DNA, and then incorporate it into another host. So when it comes to vectors, you should see that it should be small, should be compatible with the host cell in which it is going, and should not be compatible with other vectors, and high copy number, meaning when it reproduces, it should copy more number of the same kind of vectors and should be able to express itself using host DNA and RNA and should be able to move under two systems, can be eukaryotic or prokaryotic. So these are the kinds of systems that is there. So like I told you, plasmid, bacteriophage, cosmid, bacterial artificial, yeast artificial, human artificial, and retroviral vectors are there. We'll just quickly brief through this. So when you see the bacterial chromosome, what is happening is over here, plasmid is taken and then using the same restriction into basis and the phage vector is taken over here. And then you have this is the plasmid, this is the phage vector having site and a cos site. And you have an M13 phage, cosmid, and later you have a phagmid. This is the bacterial artificial chromosome and the yeast artificial chromosome. We'll just have a brief look at everything. Because time is a constraint, so I will send this particular PowerPoint to you. Just please message me in the group so that I can send the PowerPoint first and the students. So plasmid is a double-stranded extra chromosomal DNA that is present in the bacteria. It is physically separated from the bacterial chromosomal DNA and it is capable of replicating independently, usually found in bacteria. Size is around 1 to 200 kilobytes and the DNA is around 10 kilobytes in size. They have a high copy number, meaning they have a high reproducibility, and low copy number like plasmids are usually used when the protein from the clone gene is actually toxic to the cells. So different examples of plasmid are your plasmid Bolivar and Rodriguez 322, PBR 322, PUC plasmid University of California 18, F plasmid, call plasmid, etc. So you can just see. Sorry, just a second. Right. So you have an equine site, E3 site. This is the nucleotide length. The date of it is given. The different organisms it is from. It is given in different plasmids. And you get to see that this is PUC. The different number of copy numbers are also given. Here you can see that PUC and PU101 have a very good copy number. So from one plasmid, you get around 30 to 50 plasmids, which is good. So how are you going to name it? So PBR means plasmid, BR means Bolivar and Rodriguez after the, and 322 is the lab name. So you have 327 also, has an ampicillin resistance gene, EcoR1, and BAM, H1 are the sites wherein the endonucleases can act upon. Then you have a tetracycline resistance G, and then you have an origin of replication, and then after 2435. Then why are the plasmids good? Because of the small size, they're circular in nature, they're independently replicating, and at the same time, many copies are being used, and they you can have an antibiotic resistance gene incorporated in. But the disadvantages are you cannot accept large fragments. Basically, they up to 10 kilobases or kilobases can be accepted, and transformation is not very efficient. Now you have bacteriophages. What are these? You have phage lambda, lambda phage, as it is known, and M phage. These are viruses. Bacteriophages are viruses in nature. They have a hexagonal or head which has port in fact, and then this is the called the head and the collar and the tail that is there, the base plate. So upper limit is around 53 kilobases of DNA that can be accommodated in this, and the lower limit can be less than that obviously. So this is also small as such. So you have a phage lambda, a lambda phage as it is known as. Its viral DNA is around from 8.5 base DNA with 12 space single stranded sticky endomers. So this infection, what can happen is phage goes onto the cell surface receptor. Right. Now we have, so there are two different kinds of phage vectors. One is an insertion and a replacement vector. So based on the kind insertion vector, what is it going to basically take in DNA, and this can be inserted. And in case of replacement vectors, what is happening is the cleavage site is going to be one genes that are not exactly necessary for the bacterial, that is your lytic and lysogenic cycle. So you have lytic cycle that's something. So these will be deleted and will be replaced by the DNA insert. So this is how a lambda phage will be there, 48,500 base pairs. So you have lysogenic genes, and there is a restriction endonucleases which is going to remove this and replace it with the target DNA over here. And then this is going to be packaged and infected into the E. coli because bacteriophages infect bacteria. Simple. When it comes to M13, also similar in fact. You can just see the picture over here. It is having a protein code and a genome which is around single-stranded DNA superfile that is present. And you have different kinds of binding proteins and the original patient site. And then you have genes that are numbered. When it comes to phage it has an F1 origin that is imported, and it is a cloning vector which is used in combination with the bacteriophage M13. So phage can be replicated just like you do the plasmid, and it also has the DNA that is present. So two advantages over the plasmid vectors are there when you large DNA fragment. So you have the lambda phage which can insert around 24 kilobase pairs, while the plasmid vector it's only up to 15 kilobases. And it is also easier when you screen them based on the kind of bacterial colonies or the tasks that form. Whenever you use to identify whether there is a recombinant vector or not. Now we have something called as a cosmid, bacteriophage DNA that has a cohesive end site, that's the cos site. So this cos site has elements for packaging DNA into the lambda particles. So you have around 25 to 45 kilo base pairs, which is large, right? And they can just replicate just like plasmids. provided they have a proper origin of replication. And you can also have fetch capsids that are present so that you can transfer them into cells also using the method of transduction. And it is a very highly efficient method of transferring your genetic DNA through plasmids. Next is we have something called as the yeast artificial chromosome. In the yeast artificial chromosome, it has a very good transformation efficiency. In this again, what is happening is you can actually take the vectors. You have different kinds of markers also that are present, and then from the target DNA digestion with the same what do you call it, the restriction endonucleases, and then you take the DNA and try to ligate. In the case of bacterial artificial chromosome also, that is your basic coli plasmid vectors. In fact, from that you can derive the S prime plasmid having a low copy number, only 1 to 2 copies per cell. So in a cell you have only 1 copies, which can give rise to different numbers. And then usually insert size is around 150 to 350 kilobase pairs of the artificial DNA or foreign DNA you can insert into the bacterial artificial chromosome. So this is how you do. The F plasmid is cut, and then the gene of interest is also cut using the same restriction and nucleus, and then it is ligated together and then electroporated into the E. coli cells, and then you have this with the XCAL or the IPGG medium, and then different colonies wherever there are white colonies. That is when you know that it is the cells that have been transformed. You also have something called as the pi-P1 derived. So this was done in 1994, in fact. So, but then it is not exactly stable. And then when it comes to bacterial artificial chromosomes as compared to the yeast, they are stable. Transformation is easy. In fact, speed of the growth of E. coli hosts is also seen, and purification is simple. And then when it comes to human artificial chromosomes, we do not know how much of it is going to take place. These are the gene transfer methods. Can you come up with any please? So it's a tool for expression studies and determining the human function. And there is a large DNA fragment that is going to take place. But then we do not know how much of it is feasible. Now we have something called this retroviral vectors also, wherein you have the genes that have been altered, and the viral RNA is then put into reverse transcription so that we get the gene of interest. And then these, especially the retroviral oncogenes, are going to come into play over here. So how is it that we are going to choose a vector based on the amount of DNA that we want to insert into it? What is the vector's actual size? What is the site of restriction endonucleus? And after all of this, how is it going to, if at all, how much of it is going to be efficiently flown? Based on that, you have these vectors and the insert sizes. So when it comes to the making of it, you see this is the plasmid, gene of interest from here is taken, inserted, cloned into copies, and then copies of the gene are made, and basic research can be done. Over here, when we need proteins, protein expressed by gene of interest is taken and then gone ahead. So these are the references as such. Now, after this particular topic, we have seen cloning vectors also. Molecular cloning, how it is done, what are the vectors. We have seen that because 19 is the 18, 19 importance types, retrieval, plasmids, nomenclature, advantages, advantages, head, tail, we have, this is a hexagonal protein code here, patch, insertion vectors, and replacement vectors. All of this is being dealt with. Now I want to go into the final topic, that I want to teach today, which is animal cell culture. So the different kinds of equipments that we use in animal cell culture, and I just want to update it and explain it sometime so that you see cloning. Now the equipment culture in animal cell and then application of cell culture. This is also going to be dealt. How is it taking place in the animal? So fusion is a removal of the cells, tissues or the organ from the animal, and then you are going to place it in the growth media or conducive atmosphere for the cells to be growing up. So that what is the ultimate purpose for therapeutic or for to study for different diseases or to diagnose a new disease or just for the sake of research. What are the biochemical pathways and steps that are in. So when it comes to the basic history in animal cell culture, see that it was first in the embryonic chick egg cells that were placed in the saline. This was buffered by 1903, it was like Jolly was one person who had remote cells, and then he showed that they can survive and divide each with the lab conditions. In 1907, nerve fibers from a frog embryo tissue were taken and cultured into a blood clot. And Alexis Carrel was involved, he helped in developing the cell culture, and he had developed the first primary cell lines. And it does Weller and Robbins, the polio virus also. And then Eagle was one person who had developed or a particular medium, that is the growth as well as food giving and atmosphere for the cell. That is why you have Eagles, that is one thing, EM is one thing. So defined Eagles minimal essential media. Littlefield was one person who had discovered the HAT medium for selection. That is, HAT is your hypoxanthine aminopterin enzyme for growing of cells, especially the tumor cells. And then Ham was one person who had discovered the serum free media. So, what are the different kinds of equipment that is necessary in the animal cell culture to have or take? So, you have to have the biosafety cabinet. First thing, we have something called as a laminar airflow, carbon dioxide incubator, centrifuge, refrigerator, then cryo storage. In very low storage, you should put your cells and store them. And you should need a liquid nitrogen container and a heat cytometer, microscope, etc. So biosafety cabinet is also like your laminar airflow chamber basically. This is going to help you work against or in whenever there is kind of dangerous or infective microorganisms, you're going to work with them. So efficiency, they have a high efficiency particulate air filter. So HEPA filters that are present. Then you have BSC2, that is closed safety cabinet along with HEPA filters, which is around 95%. BSC3 is closed by a safety cabinet. It has a high density, you have them radioactive and sterilization, so sterilizers with efficiency of 99.5%. This is very much necessary to maintain those aseptic at the same time in different kinds of conditions, sterile conditions where you know that there is a risk of dealing with animal dealing cells that are going to be infective or carcinogenic. Incubator is one particular chamber that is going to be proper or suitable temperature, the amount of moisture that is there, pH, that is your acidity, basicity, density, all of this to be maintained at a particular temperature so that there is a very good development in cells. Sterility of it can be maintained by using HEPA filters. Constant temperature by using silicone basket is also there. Humidity can be maintained. pH is also maintained. Then you have a cooling centrifuge. That is a centrifuge which will actually separate the particles inside the particular solution based on the size, density, the thickness, that is the viscosity, rotor speed. And cooling is when it is having an inbuilt refrigerator within it, so that they not have a lot of heat generated, and that heat is going to be dissipated with the help of the refrigerator. Then you have a basic refrigerator also, wherein you store all your cells and reagents and different kinds of chemicals that is there, especially cold, where it should be maintained under less than 10, like 10 degrees Celsius. Then you have cryo storage containers, cell lines that are going to be stored, or continuous cell lines. In case there is different kind of temperature fluctuations and all, it is going to be destroyed. That case scenario, what is going to happen is passage number there. So before that takes place, your cell lines should be preserved in a cold storage container. Then we have liquid nitrogen container, which has two types of liquid nitrogen, that is the vapor phase and the liquid phase. So what is happening is this vapor phase will minimize the risk of explosion. Liquid phase is wherein you have a different kind of liquid that is present, the liquid nitrogen, and this will help in preservation of different kinds of animal cells or plant cells, whatever, and it helps freezing and preserving them in the frozen form because of its temperature minus 196 degrees Celsius. And a hemocytometer is an instrument that is going to be used to count the number of cells that are present in a particular sample and the viability of the cells. Usually, tribal test is one, Triton blue test is one, which will show you the cell viability. If at all it is dark blue, and if it is not dark blue means the cell has died and they have stayed. And the cells that have not taken up the cells are the live cells. And Yuba slide has a counting scale so that you can count the number of freezing or so flow cytometer is based upon the kind of cells that are present and based upon charge to it. It's going to be separated the cells. So you have a cell sorting machine also over here, FACS as it is known as. So this is one equipment which is necessary. And then of course an inverted microscope. I need to tell about this. Basic one for detection. You have an objective, you have a turret that is there. At the bottom, basically, you can just focus it. You have different kinds of magnification, 10x, 45x, and 100x type of magnification that is present. And then you can see, observe different kinds of cells that are present. So this is a dissecting microscope or a stereo microscope as it is known as. Low magnification, and you can actually do a dissection under this particular microscope as the name implies. You can just see, so basic ones. When it comes to the different kinds of glasswares or plastic that is there, then you have a basic beaker, pipette, then you have the measuring cylinder to know, because everything has to be appropriate. You cannot put anything and everything using guesswork. That is the reason why you need to have a measuring cylinder. And these are your T flasks, your T25, T75, T100 flasks. You have Petri plates, then you have microscopic slides, cover slips, cell culture plates, all of it that are present. So this is all for today that I will be dealing with. Next class, or that is tomorrow, I will be talking about the last three units in your syllabus. That is basically the animal cell culture as such. This block 4, that is animal biotechnology, transgenesis, and methods of transgenesis, and the different kinds of transgenic animals that are present. So yes, that is all for today. So tomorrow is going to be about transgenesis and the three chapters, that is stem cells, and one more topic that have to be covered is, just check and tell you, transgenesis, stem cells is going to be covered, and then gene therapy. So tomorrow I'll be taking around a little more time of your class, maybe extra 10 minutes. All right, thank you so much.
Excuse me, ma'am.
Yes, ma'am.
Excuse me, ma'am. Ma'am, any important questions for the exam, ma'am?
Sorry.
Important questions for the exam.
Can you please message me, and more or less, you'll have to ask in the administration for important questions. But some of the topics can be generally told, for example, different types of vectors and what exactly is stem cell therapy, all of it important questions. And at the same time, at the back of your textbook, you will have previous year's question papers, if I'm not wrong. You just have it.
Okay.
Thank you.